In this presentation, I'm going to go over how to crop and label photos, specifically for the Biology 221 lab. In this presentation, I will discuss why we want to crop and label photos, and then I'm going to show you how to do it. So let's start with a macro example of cropping, because we're more familiar with things we can see with our naked eyes, and it helps us make that intellectual leap to what we're seeing under the microscope. So here I have a beautiful picture taken out of the window of my car, and the question is, what am I looking at? It could be the mountains back here, it could be the storm front coming in, it could be the lovely sagebrush, but if I crop, or in this case zoom in, you see that there's a hawk that I was looking at. If you look over here, there he is, really little. Now, he was not that little to my eyeballs. Otherwise, I wouldn't have stopped. But this is what my camera saw. This is what my eyes saw. So the point of cropping photos that you take in lab is to get the picture to look like what you saw under the microscope. So let's go with a micro example of when you would want to crop. So here's a picture that I took using my smartphone through the oculars, and that's not what I saw with my eyes. I put the pointer on the interesting part, but this is what I was actually looking at with my eyes. See, we've got some bacteria here, and we've got a little bit of stain around it, and this is a wonderful example of a capsule stain. So by taking this picture and cropping it, you can see what I was looking at. We're going from what my camera saw to what my eyes saw. So now let me show you how to do this. I downloaded the picture that I took on my smartphone to my computer, and I named it so it would be easy to find, capsule stain bacilli. So I'm going to double click on that with my left button on my mouse, and here's my picture. So I'm going to hit open, and I'm going to open Paint. So you all have access to Windows Photo Viewer on the campus computers, even if you don't happen to have them at home. So this is really big. This is more pixels than I'm going to want, and also it's kind of fuzzy. It's kind of what we call pixelated. So I'm going to resize this. I'm going to cut that down by half. And now I've got to move my view again to get where I want. Okay, so this is much better. So I'm going to hit select, and this is going to allow me to draw a box around what I'm interested in. And then you hit crop, and you have a cropped photo. Now if you don't like it, you can always hit the undo button, but I happen to like this one, so I'm going to let's see, I'm going to save this. Now I like to keep my originals. You just never know when you're going to want to go back to that original big uncropped one. So I'm going to name this cropped. But you don't have to do this. This is just what I do. So that is how I got my cropped picture here. Now you will want to crop your pictures for your lab reports anytime you have a discrepancy between what your camera sees and what you see, or if there's stuff that's unimportant that's in the background, you may want to crop the picture. All of your photos will need to be labeled, and they will need titles. So let's once again take a macro example, and and then we will go from there to a micro example. So here I have a lovely picture. This one's a little easier to see what I was looking at. We're sticking with the wild bird theme here. So I have some wild turkeys that I saw on the way to go hiking in Utah. Now you may not know that they're wild turkeys, and because this is a science class, if you were an ecologist, you would want to know where and when I took this photo. So there's my title. It has all the important information that I would need to know for the entire picture. Now, you see that there are some rocks in here. Now, I know that these are wild turkeys and these are rocks, but if you didn't know me, you may be wondering, whether I really know which is which. So we want to label these. So I have a label for my wild turkeys, I have a label for my rocks, and I drew arrows to both of them, just so that you know that I know which is which. So going to microbiology examples, which you're actually going to be using in your lab reports, this is what I would expect for a title and a label for an auger plate, especially if it has multiple kinds of colonies. We can see that there's this kind of colony, there's these yellow guys scattered about, there's these white colonies, we've got a mold contaminant. Now there are a bunch of other colonies, but I went ahead and only labeled the ones that I'm pretending would be important for a lab report. And then I have my title that says where this sample came from, what the auger is, what the incubation temperature is. It's everything that I, as the instructor, need to know about your photo. Now let's give you an example of something that you would see under the microscope. So once again, I've got a title. I'm letting you know that this is the Staph aureus control. It wasn't a picture of my microbial pet, and what magnification I took it on. And then I also label the cell shape and arrangement. Now, I as the instructor know that these are staphylococci, but I want to make sure that you as the student know. So I want you to label these and point to them so that I know that you know. Now, I could also put brackets there or anything like that. So now let's go on to how to take my cropped photos and how to put labels on and titles. Now I'm going to do this the way that I find is the easiest. There's dozens of ways to do it. You can find your own ways. I find that it is easy to title and label pictures in PowerPoint because you can save it as a picture, and when you insert it into Word, then things don't get jumbled up when you change the formatting in Word. So I've made a new slide in my PowerPoint presentation that I've been going over, and just because this is a working slide, I'm going to change the layout to blank. Now I'm going to go over here and I'm going to click on Insert, and I'm going to click on Pictures, and I'm going to find my cropped capsule stain, since that's the one that I'm showing you all sorts of good stuff about. So I've got this. I'm going to move it to an easy spot, and I'm going to insert a text box. So I just go down here, I've clicked on text box, I go down here and I click, and there's my text box. So I'm going to put unknown bacilli with capsule, because that's what it is. Isolated from a dust sample. Let's go ahead and put originally isolated, because I've cultured this for a bit. So originally, okay, and I'm going to put that it is a capsule. Well, I guess I don't need to put that it's a capsule stain because I say that it has a capsule. Oh, let's go ahead and put capsule stain, and that it is 1000x magnification. Okay, now this is really wide and it's not particularly close to my picture, so I'm going to click on this, and by the way, I'm going to click on it so that this is a solid line, and I'm going to get so that I've got my arrows with basically an X or a cross, and I'm going to move this, and PowerPoint will provide these guidelines. Now I'm going to take this and I'm going to smush it over so that it fits underneath my picture. So I now have my title. Now I need to label this. I'm going to want shape and arrangement, and I'm going to want a label for the capsule. So I'm going to go back up here and I'm going to insert another text box. I'm going to click over here and I'm going to type in the shape. It's a bacilli, or actually they are bacilli. And so I'm going to move that over. And you know what? That's kind of hard to see. So I'm going to go over here to Format, and I'm going to change the shape to white, and I've got numerous colors to pick from, so I'm going to pick white. And same thing with the shape outline. I'm going to put black because that shows up very nicely, and I'm going to put that up here in the corner. Now I've formatted my text box, so I'm going to copy it, and I'm going to paste, and I'm going to move it over here, and I'm going to change this to capsule. Now I've got to add arrows so that everybody knows that I know that these rod shapes are bacilli and that this clear space is the capsule. Now I'm going to go in and I'm going to insert a shape, specifically an arrow, and we're going to do the bacilli first, and I'm going to draw from the box to one of these cute little rods. Now that's kind of hard to see, so I'm going to go up here and I'm going to change that. Oh, that's much easier, much easier. Let's change this so it's pointing at the actual cell. Okay, that looks good. So I've already got the color set up for this, so I'm going to copy this, and I'm going to grab this end and move it around to my box that says capsule, and then I'm going to point to the clear spot. Okay, that looks like I'm pointing to the cell. You know, this picture may be a little bit small, so I'm going to grab it and I'm going to make it bigger. Okay, now it's a little bit fuzzy, but it's going to be easier to put the arrows on there. So I've changed, let's move this over. I've changed this so it fits under the picture again. And now I've got to change my arrows. See, now that it's bigger, it's a little easier to get this arrow on the clear spot, which is what I'm after. Okay, and then here I'm going to put it actually on a cell as opposed to a clear spot. Let's click over there, and that's not real clear. Let's go ahead and move that so that it's, oh, let's go with this nice big bacilli there. Yeah, that's much better. Now, because I want to make sure, well, that's still not on the clear spot. So you gotta jiggle it, get it so that, okay, that's much better. And just so that everybody knows that I know what I'm talking about, clear space around cell. Now that's taking up a whole lot of space, so I'm gonna smoosh that down, and I'm gonna move this up. And let's go ahead and move that up a little bit. Now I could move this arrow so that it's back behind. I could click on that and send it to the back. Now it's completely disappeared. In which case I would take the picture and send it to the back. See, and then I've got my cute little arrow. So that works for me. If you wanted to have the arrow on top of the box, that's all right. Now, before I save this as a photo that I can import into Word, I need to select everything. So I'm going to come over here and I'm going to draw a box and see everything is selected. So I'm going to right click and I'm going to group these. They are now all in one group. So if I want to move this around, they all move together. Whereas before, depending upon what I clicked on, everything would move separately. So now I'm going to save as a picture. Now, for whatever reason, this portable network graphics format comes up first. I don't know what that is, but I do know what a JPEG is. And I'm going to name this as bacilli with capsule labeled. And you can name these any way that you want, making it easy for you to find it later. So I hit save, and I now have a labeled, titled, cropped picture that is ready to be inserted into my lab report. I hope that this video has been helpful. If you have any questions, feel free to get a hold of me.